advised to go to Frenchman the next night, yeah. So, I know, I didn't look again right before class because you guys know on Tuesdays I come from lab. So, I know some of you guys are unhappy with your score on this assignment. As a good student would be, right? And I didn't answer any emails because we were going to go over in class. Remember I said that? And I remember I said there was a reason why I was only going to give you one attempt. I really wanted to see what you knew. But I am going to give you an opportunity to make up points. I'm not going to open up the assignment again, because as I got to thinking about it, I'm like, that's pretty redonkulous, especially since I'm going to go over it and give you the answers. And when you got done, it gave you the answers, right? So you would definitely get a 10 out of 10 unless, you know, you have issues, right? So why not give you a different assignment, right, where you can make up your points? So that's what I've decided to do. Although I haven't posted the link, I will do that when I go back to my office. So what I want you guys to do, and this is an awesome idea that I've done in the past, is I want you guys to write questions that would be solved either by product rule or binomial expansion. Basically, you know, you can write a little story. Just change the story that's already been written. Does that make sense? Right? And if I really like it, guess where I might put it? On the test.
So you get that one free if you wrote it?
Well, if you wrote it, you should know the answer. Unless you wrote it wrong. So it'll be like a freebie. And of course, when we're taking the test, someone's going to go, oh, that's my question. Right. Okay. So I, instead of taking it here, although I could have done that, I put it into my PowerPoint that I've been building for homework because I wanted to write out some stuff. So let's go, and I wanted to take a moment and review. So I imported these slides. So yeah, I did put it sometime yesterday, huh? I don't know exactly when. But you should have gotten an alert if you have your notifications set in Canvas. If not, fix that.
Is it under module 1?
Yeah.
I didn't see it this morning.
Oh, crap. Unless I screwed up.
It's up now because I just got it.
Oh, okay. You got it? Okay.
I'm on it now. I don't see it.
So remember all the lectures outlines and PowerPoints are right here? Right. So I update this, and so it is there. Okay. And then I'll have the part 2, hopefully shortly. Okay, so this I just took from the other PowerPoint. So what do you think when you think product rule? Well, what is a product? Math class, y'all. You know the answer to this. Come on, Sterling, what's a product? What do you have to do to get a product?
Multiply.
I'll get you after. Yeah, but which one is it for a product? Multiplication, right? So you're going to be multiplying things, right? What kind of things can you multiply in genetics? Probabilities. Awesome. What type of probabilities can we multiply for the product rule? There's only a certain type that we can actually multiply one by the other. Right, independent events, right? Where one event doesn't affect the other event, right? Important. This is why I didn't just put it up there, because I want you guys to think through this and know this, because you need to know this, right? So the probability that 2 or more independent events will occur is equal to the product multiplying, right, of their respective probabilities. And the important thing here is independent events, right, where one does not affect the other. So we can't do this for stuff that we're going to start talking about in this next chapter for like sex-linked, because sex-linked traits are linked to what sex you are, right? So we couldn't look at sex separate from that trait, right? Because they're linked. They're not independent. Ooh, I just thought of an exam question. Binomial expansion, when do we use this? Well, again, what are we figuring out in genetics? Probabilities, yes, yes. Okay, so we want the probability that something's going to happen. What's the something? What are we looking at here when we're using this?
Certain genes.
Yeah, we're looking at a particular trait or gene, yeah. A genetic defect.
In like a group.
In a group, right? In a set, right? That's not ordered, right? So it's not, the order doesn't matter within the set. But we're saying you have 7 kids, right? What's the probability that all 7 are going to be boys? We could calculate this. It's actually a really low probability. But I can tell you it happened to the family that lived across the street from me growing up. 7 boys. My dad thought 3 girls was bad. Right? What's the probability you're going to have a boy or a girl? 50-50. Right? We should calculate out that just to see. It's low. I know it's low. Right? So this is unordered events within a set. Right? And so this is a really complicated equation that I promised I wouldn't make you memorize or calculate. Right? So you just got to know when you would use this. Not actually calculate it. That's not math class or calculus for that matter. That's some scary stuff. You know what gets people in calculus though? The second half of the problem. What is the second half? Algebra. And the problem is people don't barely pass algebra and then go into calculus and they go, oh, crap. Algebra's coming back to haunt me. Like this would be, another one of those like algebra nightmares we're not going to relive. So just know when we got to use it. Chi-squared too would be another nightmare. But when and why would you want to do the chi-squared test? And how would we interpret the results? Not that it's correct, but it's the best fit for what? Your hypotheses, right? What you expect to happen based on what actually experimentally happened, what you actually observed, right? So it's the statistical method for goodness of fit. What do we mean by that? How close are the observed data, right, from what you actually did for the experiment to what you predict with your hypotheses? Now, Mendel kind of did it backwards, right? He did all these crosses, and he got all this data, and then he calculated based on the data what he believed was happening, right? And he believed that you had, or they had, those pea plants, 2 discrete copies, or factors of inheritance, he called them, right? But we know them as chromosomes now, right? And genes, segments of information on that DNA that code and make something. And therefore you see this trait, this characteristic. So based on what we know, right, we can form now these hypotheses and we can test that. We can say, oh, well, we think that this particular trait is dominant or codominant, right? And we could get our pea plants and we can do a whole bunch of crosses and figure out if what we actually got matches up good with our hypotheses. So we can mathematically calculate this, right? This is statistics, right? Again, math. It does not prove that a hypothesis is correct, right? Does not, right? It just tells you that the probability of it actually being close to true. But it definitely helps us if it's really far off, to say, yeah, it's probably not the right hypothesis, right? We need to come up with a different hypotheses as to what's going on here. So you could even backpedal, right? Like Wendell did. Like, okay, well, this is what we got. So what I thought it was, it isn't, because it's not matching up. What might it be, right? And does that have a better fit now? So cystic fibrosis, we've already talked about this one, right? So this is a recessive disorder that affects the lungs, and they tell you a man and a woman are both unaffected heterozygous carriers for cystic fibrosis, because remember, it's recessive. So both, you have to have both recessive, basically non-functioning genes to have this disease. So we know, though, the bad news with the heterozygous parents is they both carry that recessive trait, right? But they also both have the dominant trait, so neither one of them has the disease, right? They produce enough functional protein, they're okay. So the question is, if the couple has 2 children, what is the probability that their first child will be unaffected and their second child will be unaffected? So basically both their kids will be unaffected. What are we going to use for this? Product rule, chi-squared, or binomial expansion? Okay, so how many people say product rule? How many say binomial expansion? Because that's the other one I heard. Okay, the product rules have it and the product rules have it. That is the correct answer. But let's talk about why that's the correct answer. And the chi-square was definitely not it, y'all, because remember, when I assigned the assignment, I got tired. I told you guys I didn't make it to chi-square, that there were no chi-square. If you were listening carefully. Okay, so I intended but didn't get there. We will get there. So why? Why product? So someone who raised their hand for product, explain why you picked product. Somebody raised their hand. Okay, Angela. Right, they're independent events. So did anybody get tripped up by the fact that they said first and second? Okay, no, good. All right, so they're independent events. Does that make sense? So that's why we can multiply them. Okay, so let's walk through this one. So what's the probability that the first child is unaffected? Well, first we got to do what? A Punnett square. But we know this Punnett square like by heart at this point, right? 2 heterozygous, it's easy. Walk it. Just change the letters. But there it is, right? So 2 heterozygous, you're going to have 3/4 or 75% unaffected, and the sad affected is going to be about 25% or 1/4 out of your probability, right? Still with me? Okay. So some people like percentages as opposed to fractions, right? Some of us consider fractions evil Siamese twins. Because, you know, if you don't do the same thing to both sides, they get mad. Because they're connected, numerator and denominator. But can you multiply percentages? Yes. If they're in what form? Decimal form. That's the trick. Decimal form. Okay. So I think this one, for one of the ones, I pasted in like the copy answer from the book, and they did it in percentages, but notice they were in decimals. Right? Either way, you're going to get the same correct answer. So what's the answer for this? What's the chance you're going to have an uninfected child? 75%, or 3/4. Everyone still with me on that? All right, easy peasy. So the next one is basically asking the same thing again, is it not? So, except this is the second kid, right? And so the second kid, what's the chance? The same, right? Because they're not connected. So 75%, or 3/4. Still with me? So to actually calculate it, which I'm not going to make you guys do, you'd multiply 3/4 by 3/4, or 0.75 by 0.75. And you would get 56%. So you have a 50% chance that both your kids would not have cystic fibrosis. So now, if that couple, that same couple, who are both unaffected, have an affected child, what is the probability their next child will be an affected girl? So first question is, what are we doing?
Binomial expansion.
Why binomial? Yeah, but those are two separate events, are they not? And they're not in a set, right? We're not saying, what's the chances that you're going to have, 3 out of your 7 kids have cystic fibrosis, are we? So this is still product rule, but yeah, we're looking at 2 things. We're saying, is it going to be a boy or a girl, and is it going to have disease? So it's what? Product rule. So then I broke it up for you guys. I said, what's the chance they're going to be affected? This is heterozygous. What's the chance they're going to be affected? 25%. What's the probability you're going to have a girl? 50. Oh, I didn't put the answer in. What's wrong with me? That's okay. 1/2 times, or 50% times 25%, whatever that is. I don't know. Yeah, not worried about it. I don't know why I put that one in there, though. Paste and copy didn't work. All right, so now on to another disease. Except this one is not so great. And this is a real thing as well. So Huntington's disease, which is on chromosome 4. So on my poster in the room, you can find it listed on chromosome number 4. It's a rare dominant trait, which means that if you have the defective gene, you have the disease. Just one chromosome, one defective copy from one parent, you have the disease. So heterozygous individuals have this disease. Homozygous definitely have it. And this is where this one can be kind of tricky. Yeah, you're jumping ahead. Okay. It's okay. It's okay. So a man in his 30s who already has 3 kids. Okay, so he's already had them. He's in his 30s because this doesn't manifest until later in life, typically, between 30 and 50 years old. So people have usually already had kids and then, oh crap, they come down with this disease. Discovers his mother has the disease. Okay, so she's older, maybe in her 50s, right? She's got the disease.
It's not as sex-dependent.
No, it's not sex-linked. Yeah, not sex-linked. Good question. We haven't got to those yet. That's this next chapter. So they're asking, what is the probability that the man in his 30s will develop this disease? Well, the question is what is his mom, right? We can speculate. And that's the one thing I didn't like so much about this question. Is you guys didn't get much background on this one. So the possibility, right? Dad doesn't have it, right? So he's probably recessive, homozygous, right? Mom has it. So she has at least one of the affected dominant traits, denoted by the capital H here. She could be homozygous or heterozygous. Which do you think she probably is, since this is rare? And that was the key there. Heterozygous. Heterozygous. Because think about this. If people were homozygous, all their kids would have the disease. And there probably might be some people out there that are homozygous. More often, you're heterozygous for it. So if she's heterozygous, what's the chance that he's going to get the disease? 50-50. It just depends on which one she gives him. If she gives him the dominant one, he's going to have it, right? If he ends up heterozygous like her. If he gets the good one, from both parents, then he's A-okay.
So how are we supposed to know that rare means heteros? I mean, is that just
That's why, again, like, I realized after the fact that it wasn't enough information when I worked through the problem.
Well, we got it wrong here, like,
No, did you come in late today? Yes, you did. All right, I'll repeat later.
Plus 50-50.
It's 50-50. It's 50-50.
Back to the question I asked last week. So it is possible when we say about the recessive, like both parents could have the disease and it's skip the kids and show up in the grandkids or whatever.
Right, yeah. And again, it has to do with the pattern of inheritance. So, we had to figure out the probability of the dad is 50%, right? That he has it. So now what's the probability? It's basically the same situation, right? Him and his wife, as say maybe him, his mom and him, right? What's the probability that he's going to give it to his daughter? He's probably heterozygous, right? And so is his mom. It's 50-50.
But he's 50-50.
Right.
So he might.
So the first question was the probability the first child, right, will get it is 50-50. But she's right. And the dad is 50-50, right?
It's the same kind of square.
Yeah, but we basically have to do product rule here, right? We have to multiply 50% by 50%, which is 25%. And although I promised I would make you guys do it, I kind of did. But product rules are easy. Binomial expansion is the nightmare. Is everyone still following with us? Okay. So what's the probability that one of the three of his children will develop Huntington's disease? So what would we use to solve this one? Right, this is a set, so this is binomial. Does everyone understand why? Okay. Yes. It's a set. That's the clue. Alright, so then I asked you guys another question, totally not related. A true breeding yellow plant was crossed to a green plant. Yellow is a dominant trait. The first generation individuals were allowed to self-fertilize. What is the probability that for any 7 plants, there are yellow and 3 are yellow and 4 are green? I didn't ask you to calculate it. I'm saying, how would you solve this? Binomial, because this is what? A set. So, how can you gain back points you lost? I'm going to set up a Canvas assignment where you can basically paste in, copy in from a Word document, where you write, and I'll give you 1 point for each one of them that's good up till the number of points to bring you to a 10. So for instance, if you got a 5 out of 10 on this quiz, right, and you need 5 points for your perfect 10, then you can write me 5 product rule or binomial expansion, a mixture thereof, questions. You don't have to necessarily solve them. Okay? Next Tuesday. 1 week from today. So you got plenty of time to come up with your cute little stories. Right? Doesn't have to be real stuff, but real stuff is cool too. Just make sure you give us enough background information, something we didn't do with the Huntington's disease, right, for these questions. And you could be the lucky winner that yours makes it onto the test. And therefore, you should definitely know the answer. Anything about those instructions not make sense?
So we're just writing to where the answer would be like.
Yeah, but you see, yeah, so you would write out, say, this question, like just like this, but you need to tell me which is the answer and why, too, right?
Okay.
No, just guessing. And you can't copy any of the ones from the book. Not word for word, anyways. Change them up. So, we could change this to a true breeding white cat breeds with a true breeding black cat, and black is dominant. And the first generation, what is the problem, right? You see? It's not hard. You just take some effort and creativity, something I sometimes lack. Okay. We good? All right. So I'll set that up, and you guys will have until Tuesday to post that. So I haven't done any chi-square problems for you yet, so I will hopefully get that done also by Thursday, where you guys get some practice with that. Those are a little involved, so again, I lack creativity. So I usually just modify the ones from the book in the homework. But I got to put it into Canvas for you guys, right? Because I'm not going to make you calculate out the chi-squared. But I'm going to have you interpret them. So, as we discussed earlier, right, under Module 1, under the link for outlines and lectures, Module 1, I added just the first part of Chapter 4, which is what we're gonna work on next. And I should have the second part up shortly. So now, we've been talking about Mendel's laws of inheritance, right? 2 laws. What's the first one? Law of, one's independent assortment, the other one is segregation. So what do they mean? So tell me a story, right? Based on how you get your stuff from your parents, what tells you that they either segregate or independently assort? Who thinks they can tell us? One or the other. Go for it. So your eye color is determined by what? Which you get from your parents. So eye color is determined from your genes that you get from your parents. Both parents, right? Okay. Which they got their information from their parents, right? So how many sets of chromosomes do they have? They have 2, right? One from their mom and one from their dad, right? What happens next? So they've got these chromosomes they got from their parents, and they give them to you, right? But does your mom give you all of her chromosomes? No, she gives you half, and your dad gives you half. Now, of course, they got their halves from their respective parents, right? So how is it determined what chromosomes, what half your mom gives you? Dominant determines what the trait looks like. Is there anything that determines which chromosomes your mom got from her mom and her dad she gives you? No, it's totally random. Right? It's totally random. That's what that law means, is that you get from your parents what they got from their parents randomly. It gets mixed up each time. Sometimes not so mixed, right? Me and my mom look like the same person. My sister, on the other hand, doesn't look anything like us. There was a whole lot of mixing going on there. Does that make sense to you guys? Okay. independent assortment doesn't always happen, right? Independent means one thing independent of another, right? So this is when we start looking at dihybrid crosses, or even more, like when we're looking at two traits at the same time. Are they going to independently separate from each other? Yes, if they're on separate chromosomes. And Mendel got lucky with his pea plants. All 7 of those characteristics he looked at were on different chromosomes. Are we going to continue to talk about those types of things? No, we're going to talk about stuff that's linked, right? Like things that are on the X and Y chromosomes. So therefore, depending on which you get, X or Y's, determines whether you have or don't have a particular disease. So hopefully we're building on our understanding of segregation and independent assortment, right? So this is, and so far we've just looked at a single gene that had 2 possibilities. Although we did look at one that had multiple possibilities and already kind of pre-looked at codominance, right? When we looked at blood typing. Because again, I wanted a real world example that you guys could link to. Other than that, other than the blood typing, we pretty much stuck to just dominant recessive relationship. In this chapter, we're going to go beyond that. We're going to talk about stuff that deviates from that simple plan. They still obey some of the laws, just however, a little bit more complex. We're looking at more stuff. So you need to be able to compare and contrast the different types of inheritance patterns. And you need to be able to describe to the molecular level what these differences are. So this table I know is like, this table, which lists all of them for this chapter. Is like microscopic, right? But you know, this is an excellent like summary, right? And that's what it is, or preview, right, to what it is that we're going to go over. So back up for a second. So why do we look at these things? We have 2 things in mind. We want to be able to predict outcomes, right? If we know how these things are inherited, we can predict, right, what's going to happen. And this is important as it comes to certain genetic diseases, right? We want to be able to predict out, you know, what's the chance our kid is going to have Huntington's disease, right? And help us, of course, understand the relationship between the molecular expression, right, actually making those proteins that actually do something that give rise to that particular trait. So there's our big table, right, which talks about what that inheritance is and then what is happening at the molecular level that gives rise to what we see. So we're going to start out with some important definitions. I've said this one once before, right, but we're going to start using it a little bit more. What is a wild type versus genetic pleomorphism? Why loss of function are typically recessively inherited. And how alleles can exhibit incomplete penetrance and how they vary in expressivity. So these are all terms that are probably like, right? So we're going to break them down. So when we look at a whole population, right, what is the most prevalent characteristic? What are those differences? That is referred to as the wild type. typically these are functional proteins. They function normally, right? Which is why they're the most predominant, right? Because if they weren't functioning normally, they wouldn't be beneficial to have, probably, right? And so not everybody would have that. So most of us have the normal functioning hemoglobin, right? It's the non-functioning sickle cell hemoglobin that's not as prevalent because it's abnormal, right? Doesn't function right. You're at a disadvantage if you have that. Remember that you have DNA, and that DNA is making something. So in the case of hemoglobin, right, it's making that protein to carry oxygen. The question is, if you only have one good functional copy you got from one parent, is that enough? Can you make enough of that hemoglobin protein to get by, to be okay? So we can make enough. Genetic pleomorphisms can produce more than one wild type in large populations. So, I guess we don't have an example for this one just yet. They give some flower example, and I can't remember it off the top of my head in the book. But to go back to, say, blood type. A and B aren't really all that different. And they're both dominant. We talk about codominance with them. So this is two choices. They talk about some plant and having two different colors, right, is common. Right. Alleles may have been altered by mutation, and therefore we term these as mutant alleles. So that means that when the information was being copied, it wasn't copied correctly. That's what a mutation means. And that was passed on, right? It wasn't fixed. That messed up copy is given to the children. That's how these, you could say, defective genes have come about. So, because they're just that. They're defective. They don't do what they're supposed to do. They don't make a functional protein. In the case of sickle cell, the hemoglobin is functional. The problem is that its interaction with the other hemoglobin in the cell causes to aggregate in a way that deforms the cell. And then therefore decreases its effectiveness. These tend to be rare in populations, again, because if it's defective, if it's not working properly, you know, you're not going to predominate in the population if you have a defect. So these tend to be inherited in a recessive manner, where you have this problem because both of your genes are defective. Right? So full-blown sickle cell anemia, you got the bad copy from both parents, right? This is recessive. So lots of the diseases that we look at, right, follow that type of inheritance and have been linked to a mutation, where there's been a change in the genetic code such that now you have non-functional proteins. And so therefore, again, it's usually a recessive manner, meaning that as long as you have at least one good copy, it's enough that you're not going to have the disease. So, we talked about PKU, right, where they can't metabolize phenylalanine. Albinoism the lack of pigmentation in the skin and the eyes and the hair is a defect where you're not making a protein that contributes to the production of pigment in those areas. Tay-Sachs, which is very common in the Jewish community, because again, people kept breeding with each other, and you end up with this concentration within that population of this particular disease because of inbreeding. This is another enzyme that doesn't work properly, and it can lead to paralysis, blindness, and eventually death. Another similar enzyme, another defective disease with lipid metabolism, cystic fibrosis. It's the chlorine. Remember, I can remember what ion? It's the chlorine transport ion that they have a problem with. I don't expect for any reason for you guys to memorize any of these, right? This is just give you an idea of, just a few. That poster over there lists a gazillion different diseases linked to specific mutations on specific chromosomes. Because like I said, Huntington's disease is on chromosome 4, so you'll see it listed on the 4 chromosome. Some of them we know, the human genome has been mapped, right? We know where most of these defects are. And right down to the molecular level of what nucleotide do you have wrong, right? So in the case of my husband, it literally is one nucleotide wrong. One little AGTC, and he has a disease. Hemochromatosis. So a simple dominant recessive relationship. The recessive allele does not affect the phenotype of the heterozygous. Why is that? So this means you have one good copy and one defective copy. But you don't have a disease. You're fine. Why is it that you're fine? You got a bad copy. But the good news is you have a good copy. And that good copy is enough, right? What we call dominant, but in truth, it's enough to take care of you, right? So there's 2 possible explanations. 50% of the normal protein is enough, right? So just having half normal protein, you're good. What's another possibility? That you only make the normal protein and you don't make the defective one. Or you do what's referred to as upregulation. So mostly you are using the functional protein copy, that chromosome, for the most part, right? The body realizes this other one, defective, let's not use that quite so much because it doesn't give us anything good, right? We have a way of regulating and saying, okay, let's just use the good copy. We're just going to make more copies of the good copy, right? We're going to make more protein from this one good copy that we got. Make sense? So for some diseases, again, we know, what's happening, right? 50% is good enough, or we upregulate. Does that make sense to you guys?
Can you explain that number 2 again?
Okay, so there's 2 copy machines in our department, right? One of them likes to eat paper. He's hungry, apparently. And the other one pretty much works good all the time. So if you've got 10 minutes to print your test, which one are you going to use? The one that doesn't eat paper, right? The good one. You see? So you're going to avoid the bad one, right? Because it's not producing what you need. You're going to use the good one that's going to give you what you need. You're going to upregulate. You're going to use the good one more often, right? Only out of desperation, which in the case once you figure out it doesn't really work, you just pretty much don't use it.
So is that like the difference between some people who suffer more of the same disease?
Yeah, because the thing is, it depends on the disease, and there's lots of complications, lots of factors that could factor in. But it could be at the core, yes. So her question was, is this why some people who have the same disease are affected differently?
Mm-hmm.
It could be, but it's not that simple usually. Yeah, it could contribute for sure. So, which is determined by your genetics too, and the environment you live in. Right? Lots of factors, lots of variables. We wish it was like so simple, right? But it's not. Okay, so, but for purple color in the pea plants, it's pretty simple, right? That we know the heterozygous, right, is still purple because it has one good copy that makes that purple protein. And that is enough to make that flower purple. Just as purple as the one that has two good copies. 50% of that production is enough for purple. White is actually a loss of function. It doesn't make any pigment. So it's white, right? It's recessive, two defective copies, doesn't make pigment, doesn't have color. What happens when you have a dominant mutation? These are much less common than recessive, right? This is like with Huntington's disease, where there is a change that has happened, and that protein or that product is expressed in such a way that it's damaging. There's three possible explanations for this. Gain of function, where the change caused it to be able to do something it never used to be able to do. So the body doesn't know how to regulate this. It's like, oh, well, okay. Why are you doing that? And I don't know how to stop you from doing that. It's apparently not good, but we don't have any way to do anything else. So in this case, the protein encoded by the mutant gene is changed so that it gains a new or abnormal function, right? So this is something new. This is out of the norm. And unfortunately, the reason why we're talking about it is because it's bad, for the most part, usually. So Huntington's disease is an example of that, where it changed. It's not non-functioning, instead it's doing something it shouldn't do. Dominant negative, what we mean by that is the protein encoded by the mutant gene acts antagonistically to the normal protein. So this is where, if you're heterozygous, right, you're producing the normal protein, but you're also producing this other protein, this mutated protein, who apparently wants to fight with the good guy and stop the good guy from doing his job. Not good. Most women know about an interesting antagonist as it relates to birth control. What other medication do you not want to take when you're on birth control? Because it's antagonist. Antibiotics. Antibiotics are antagonists to most birth controls. And so in other words, they fight with the birth control, and the birth control doesn't work anymore. And that's how you sometimes end up with babies.
I learned that it was it's oral birth control. If you take oral antibiotics, it's because, like you said, they fight. But if you have like an IUD or a NuvaRing, it doesn't affect it.
Right, it's different.
That's not true.
Oh, yeah, well, the thing is the way an IUD works is it's both physical and usually chemical. So the chemical aspect will probably still not work because, again, it's two chemicals fighting against each other. But the physical aspect would still work. to a certain
If
extent.
I got pregnant with my daughter, I actually took the shot and was on, antibiotics at the same time. So
And they didn't tell you.
Mhm.
Or in the case of one of my nieces, they forgot. They're like, oh, we probably should have done that without protection. And there came Carly. So, God wanted it to be. This next one, haploinsufficiency. So the mutant is a loss of function, right? So it's mutated, it's not doing what it's supposed to do. The problem is if you're heterozygous, it doesn't make enough product to give you the wild type phenotype. So we don't upregulate this one, right? So you got a bad copy, it doesn't make what you need. Your other copy's good, but just having one is not enough to make all that you need. There's no upregulation on this one. So you end up with half of what you need. It's like going to the checkout counter and you got $20 and the bill comes to $25, and then you're figuring out what you're not going to buy. Put this back.
So, when you use the word dominance in reference to this, you're really just saying that that's the trait you're going to display?
Yes. So in the case of Huntington's disease, you're going to have that disease if you have that one defective copy. And in that one, I think the problem is it's doing something it's not supposed to. It's like rogue. Don't quote me on it, though. Yeah, I think that one's a gain of function. But again, don't quote me on it because I haven't looked it up. Okay. So the next is incomplete penetrance. So this is where the dominant allele does not influence the outcome of the trait in a heterozygous individual, right? So they've got two different copies. The dominant, right, is not actually showing the trait. So it's not making it from the DNA level to the cellular or organismal level, right? We're not seeing the effect of having that dominant trait. So imagine this is like having the gene to make purple pigment, but you're a white flower. What the heck? What happened there, right? You have it, it's dominant, it gets expressed. Why aren't you purple? Well, in that case, purple, there wasn't enough purple made, right? It's so little, it looks white. So the example they give here is polydactyly. This is a dominant trait. So if you have it, you should end up with extra fingers or toes. Autosomal means it's not on your sex chromosomes, it's on all the rest of them. But it's dominant. It gets expressed. Affected individuals have additional fingers or toes. A single copy of polydactyly allele is usually sufficient to cause the condition. Right? So you have one, you're heterozygous, you have one copy of the defective dominant defect. You end up with extra fingers and toes. Although some individuals don't end up with extra fingers and toes. Whoa, what's going on here? Right, so look at a family tree. Extra fingers and toes. Extra fingers and toes. This one inherited it, right? Because look back, right? Somebody had extra fingers and toes in the family. Didn't have extra fingers and toes. Spouse didn't have extra fingers and toes. Oh, kids have extra fingers and toes. Where did that come from? Right? They have it, but even though it's dominant, they're not expressing it. It's not penetrating all the way through to the organism level. Why would that be?
It's not enough protein.
I don't know why they picked this one, because I wish I would explain it out. Because I'm not sure, like, I think there are other factors there that kind of will sometimes shut this one down. But I don't know if they know all the inner workings with this one, but they definitely know that it's dominant. But yet, there are people that have it, that are heterozygous, that will not display it. So when we look at this as a population, right, we can figure out how often it penetrates through, right? So for 60% of the heterozygous that have this dominant trait actually end up with polydactyly. Any individual either has it or doesn't, though, right? You either have extra toes or not, as far as the expression of the trait. This one also goes into the level of expression. So this is where some of you guys were going to. Well, somebody has the disease, why are some people more affected than others? And we refer to this as expressivity. The degree at which it's expressed. And again, so someone who has it may end up with one extra finger on each hand, where someone else in that family ends up with extra fingers and extra toes. Why did one of them only end up with extra fingers and not extra toes? Yeah. It's polydactyly. So if you have, of course, it highly expressed, you have lots of extra digits. If it's not as expressed, you have a lower number of extra digits. The specifics into this, for some of these things, is not well understood still. This is the terminology we use to explain it, though, right, or to talk about it. So which of the following is not an example of a wild type gene? So yellow flowers in the elderflower orchid or red flowers in the elderflower orchid. Well, this is that example I was trying to remember. In that population, yellow is about just as common as red, right? The albino elephant, right? That's rare, right? Usually they're gray. Right? That's the wild type that we see. And again, the albino is a defect, right? It's a loss of function and a recessive inherited, so it's very uncommon. It's rare. It happens, though, right? We've got the albino alligators, too. So dominant alleles may result from mutations that cause let's look at each one of these as a true and false statement. The overexpression of a gene or its protein product. What do you think? Okay, some people think true, anyone think false? Okay. A protein to inhibit the function of a normal protein. True, right? This is the antagonistic relationship, right? A protein to be inactive and 50% of the normal protein is sufficient for a normal phenotype. True. So, therefore, all of the above are true. Remember to always keep reading. These were the three that we talked about, right? Polydactyly is a condition in which a person has extra fingers and or toes. It is caused by a dominant allele. If a person carries the allele but does not have any extra fingers or toes, this is an example of incomplete penetrance, right? Didn't penetrate all the way through. What do we have left? And see, I thought I put too much. Oh, good. Wait, we're going to get through this last one. Just because it's a lead into the next one. So the molecular expression of expressivity and incomplete penetrance may not always be understood. In most cases, the range of phenotypes is thought to be due to influences of the, what do you think? Environment is definitely one, right? And or other modifier genes, right? So other genes that may take care of the problem, right? Or stop it from being expressed, which I think is the case definitely for polydactyly. I think there are other genes that when that one is defective kind of can make up for it being defective. So for the next one, we need to be able to discuss the role of environment with regards to individual traits and define the norm of reaction. So these are important to some of our next things we're going to look at. So environmental conditions may have a great impact on the phenotype of an individual. So I gave you guys the example of PKU before, right? You could have this defect, and as long as you don't eat what, you're going to be fine. Phenylalanine, which aspartame has a lot of, right? So, you know, this is one of those things where the environment could really make a big effect on the individuals. The example they talk about here is the Arctic fox, right? Its coat color in the different seasons is dependent on the environment, on the temperature. And that affects the expressivity of whether he's going to be his brownish color, right, or his white color in the winter months. So he changes, and it's temperature dependent. And then there's the PKU, which we've already talked about. So this one's interesting, where the environment will affect the number of facets. So little facets of the eye of the fly. They've done an experiment, and it's based on temperature. So at 15 degrees, they have over 1,000 facets. But at higher temperatures, they have around 150 facets. Does anybody know in the South, one of the things that we talk about as it relates to reptiles and temperature? The alligators, the temperature in the nest where the egg is developing determines whether it's going to be male or female. Oh, I don't remember which is which, but I know temperature determines. It's a certain cutoff, yeah. It's interesting. Some of them maybe. But I know it's definitely common in reptiles. I know it definitely happens for alligators. So what's the normal reaction? This allows us to see, right, the effect that the environment has on the phenotype. So it's one of those weird terms. I don't know how best to define it. So the outcome of an individual's trait is controlled by genes and environment. I didn't have an answer slide for that one. And I actually ended early. By like 3 minutes. So be working on those problems, questions, right, for submitting, and start reading ahead in Chapter 4. I'll post the rest of Chapter 4 PowerPoint, and we'll pick up from where we left off.